Okay. Hi, everyone. It just hit 1:33 p.m. UK time on my watch. Hello, everyone. Thank you so much for joining us for the second edition of the GSU Malaria Journal Club monthly series for 2025. Today's webinar is a vector-focused one, referencing Sophia Mwinyi's recent preprint titled Genomic Analysis Reveals a New Cryptic Taxon of Malaria Vectors with a Distinct Insecticide-Resistant Profile in the Coast of East Africa, as you can see on the screen. My name is Pablo Chauke, for those of you that do not know me, and I'm a training coordinator at the Genomic Surveillance Unit, working with an awesome team of people, some of whom are in the room right now, to organize these monthly events. Just a few ground rules. The Journal Club is being recorded and will be available on our Malaria Gen YouTube channel later today, so please do check it out. You can rewatch the recording and share with your colleagues and your networks as well. Kindly stay on mute so as to not disturb our speaker. We will have a Q&A session at the end where you can raise your hand or type your question in the Q&A or chat box. So you'll be able to raise your hand and we can allow you to unmute and you can ask your question. But throughout the presentation, please kindly type your questions in the Q&A and Sophia will address them at the end. Without further ado, I would like to introduce our speaker for today, Sophia Mwinyi, who has been an integral part of our flagship introductory vector training course as a teaching assistant. And yeah, we've known her for the past 4 or 5 years, if I'm not mistaken, and it's been a great relationship working with her. The title of the talk is similar to the preprint, as you can see on the screen right now. Just a few words to introduce who Sophia is. Sophia Mwinyi is a research scientist at the Ifakara Health Institute and a PhD candidate at the University of Glasgow. Her research focuses on leveraging innovative technologies, including machine learning and genomic data science, to enhance vector-borne disease surveillance as well as control strategies. Sophia Mwinyi's current work focuses on the application of machine learning, infrared spectroscopy, and the genomics data science to track key biological threats in malaria control. Beyond her contributions to malaria research, Sophia co-leads a groundbreaking project exploring the potential use of machine learning as well as infrared spectroscopy techniques to detect volvulus in black flies, the vectors of river blindness, a neglected tropical disease that continues to afflict thousands across multiple countries in Africa as well as South America. Sophia, thank you so much for joining us. You may begin your presentation.
Thank you, Pablo. Hi, everyone. So as Pablo has already introduced me, today I'm going to present to you a work that I've been involved in. And currently it's a preprint that is awaiting publication, titled Genomic Analysis Reveals a New Cryptic Taxon in the Anopheles gambiae complex with a distinct insecticide resistance profile on the coast of East Africa. So to begin with an introduction, this work particularly focuses in Tanzania, and Tanzania is one among the countries that is highly affected by malaria. Tanzania has high morbidity and mortality of malaria. According to the WHO World Malaria Report from 2024, Tanzania ranks the 6th highest with malaria cases, while it's the 4th highest with malaria death. And in 2022, the average prevalence rate of malaria in children under the age of 5 was 8%. But as we can see, that the malaria prevalence in the country, in Tanzania, actually differs by regions from the map here. And we can see like some regions on the northwest part of Tanzania, the upper part here, has high malaria cases, as well as the southeast part, while the middle part of the country actually has very low malaria prevalences. So we're going to see the major reason for this is highly driven by the climatic differences or climatic, climatic differences. So from the Köppen climate map, we can see that the upper part and the lower part of the country actually are characterized by tropical, as well as, tropical climates, whereas the middle part is characterized by the semi-desert and, semi-arid and arid climate. So the upper part and lower parts have a higher prevalence, given that they have a very conducive environment for malaria vectors to breed and to reproduce as well. Whereas the middle part is arid or semi-arid, which makes it very difficult for malaria vectors to exist. So the main objective of the study was to use whole genome sequence data from MalariaGen, which is an open source repository of genomic sequence data of vectors and parasites. But particularly, I was looking at the Anopheles gambiae complex mosquitoes that were collected from 4 sites in northwestern and northeastern parts of the country to address 3 key questions. The first question was looking at the taxonomic status of Anopheles gambiae complex that were collected from the country. The second question was to determine the status of geographical structure of the present species of the present species that were identified from the first objective in different parts of the country. And then the third objective was to determine insecticide resistance profiles of the existing species and the possible spread of resistance across geographies and within and across species within the complex. So to begin with, the map here shows us the locations where our data was collected. And we see we have 4 main locations where we collected our data. And these areas include Muleba, Tarime, Muheza, and Moshi, whereas Muleba, Tarime, and Muheza, they have an equatorial climate. We see Moshi has an arid or semi-arid climate. And the table here shows us the distribution of our mosquito samples by species, as well as by years of collection and locations. And the samples that we collected from Tarime and Moshi were collected as larvae, and they were raised to adults in the insect trees. And also the samples that were collected from Muheza and Muleba were collected as adults using indoor resting collections and pyrethrum spray catches. So to address our first objective, we used ancestry informative markers, which is a method that uses genomic markers from a specified number of species. And in this case, we used the ancestry informative markers were based on Anopheles gambiae, Anopheles colutzi, as well as Anopheles arabiensis. And the method actually informed us that there were 3 main species, that is Anopheles arabiensis, Anopheles gambiae, and an unknown species group. However, given that this method can be restricted, given that it only uses a limited set of genomic markers from selected number of individuals, it actually has a problem of not being able to capture the full genomic variation across our entire sample space. And so we went on to use a different methodology to ascertain our results, and that is the principal component analysis. And this table here actually shows the results of our principal component analysis, where we have 4 main clusters. The first 2 clusters are actually, they only constitute of Anopheles gambiae samples, but we can see here, this cluster particularly included only Anopheles gambiae samples from Muheza, whereas this cluster constituted of Anopheles gambiae was constituted of Anopheles gambiae from Muleba. And then we see all the Anopheles arabiensis samples across the country, that is from Moshi, Tarime, and Muleba, were all clustered within one group. And we have a fourth cluster where we see a number of unknown species, as well as the Anopheles gambiae samples that were identified by ancestry informative markers. But as I've said earlier, that the ancestry informative markers methodology is limited, and so it may likely not also be able to distinguish previously unknown taxa or like species. Yeah, it might not be able to distinguish, to distinguish the previously unknown taxa from the existing ones. And we see that there, whereas like from the previous map, we see that there is aridity through the central corridor of the country, where also the Rift Valley passes, and so we see that this may prevent the mixing of Anopheles gambiae populations, and that's why we can see there are two clusters of this particular species, whereas for Anopheles arabiensis, we don't see any of this separation. So we also, on top of that, since we found some unknown species, we also went on to integrate data from outside Tanzania, including Kenya and Uganda samples. And we used a new method that is very similar to PCA, that is the neighbor joining trees. And from the neighbor joining trees, we can see that we see similar results to the PCA, where Anopheles arabiensis samples from all the three countries are grouped together, whereas we get also two main groups of Anopheles gambiae. And the first group of Anopheles gambiae, that is the samples from Muleba, from Tanzania, were clustered together with all the samples from Uganda. Whereas the samples from Kenya were clustered together with the Anopheles gambiae samples from Tanzania in Muheza. And also we see there are two different clusters here of the unknown species. That is, the first cluster is from Tanzania, and then other clusters from Kenya, and they're closely related. And so our study identified that there is an unknown, the same unknown species that we find in Tanzania, we also find it in the coast of Tanzania, we also find it in the coast of Kenya. So the main question is it any of the taxa of the taxon in the complex other than Anopheles gambiae, Anopheles arabiensis, or Anopheles coluzi, given that Anopheles gambiae complex has seven other species? So we did a neighbor joining tree analysis, and from this we included samples that we included data that included most of all the other known species within the Anopheles gambiae complex, that is Anopheles melas, meras, quadriannulatus, and coluzi, as well as gambiae and Anopheles arabiensis. And we can see here that the unknown species still clusters separately from the known species. That is Anopheles arabiensis. You can see it's still on separately clustered, whereas we can see gambiae here that is closely clustered with Anopheles coluzi, and then we can see the Anopheles meras is on its own, as well as quadriannulatus and Anopheles melas. So, given that the two groups of the unknown species are found in the coast of Kenya and Tanzania, we also know that Anopheles meras also exist on the coast of east to southern African countries. And so these results actually show us that this particular group, despite it being found in the coast of these two countries, it is not Anopheles maris. So we went on to do another analysis where recently there have been studies that have found new cryptic taxa in West Africa. That is the Gondry and Tengrela from Burkina Faso, as well as Anopheles fontanile from Gabon. So we wanted to see whether these the unknown species that we are seeing from this study are associated with the recently cryptic taxa that were identified from West Africa. And from our analysis, it shows that these two groups of the unknown species are actually not associated with any of the recently identified cryptic taxa. That is fontanile. We can see it's clustered together here, but it's separate from our samples, as well as the Gondry and Tengrela, which are clustered separately. We went on also to do the FST analysis just for the samples from Tanzania and Kenya. And from this, we see that there is a very high FST between this group, the two groups from the coast of Kenya and coast of Tanzania with other species, as you can see here. But we also went on to do another analysis on admixture, and we saw that there was no evidence of admixture between these species. And so given that our two groups were found in the coast of Tanzania and Kenya, we designated this particular unknown species as Pwani molecular form. Pwani standing for coast in Swahili. So went on to look at the geographical structure of our samples, and from the geographical structure, we particularly did the genetic diversity analysis, and we find that there is a similar genetic diversity across Anopheles arabiensis samples, as we can see here, across all the three estimators. Whereas there is a distinct genetic diversity between Anopheles gambiae mosquitoes collected from Muleba and Muheza. This shows this coincides with our results from the PCA, and this is likely due to the fact that Anopheles gambiae they prefer more humid areas, whereas Anopheles arabiensis are they're more adaptable to different climatic conditions, and so we can see that they are very likely to like continue breeding and to migrate from one area to another and still continue to exist. And so there is no restriction of gene flow within the species of Anopheles arabiensis, whereas in this case, given the presence of the arid area in the central corridor of our country, as well as the presence of the Rift Valley, we see that there is a separation between the Anopheles gambiae from the western and the eastern side of the country. The Pwani molecular form also, we see it's very distinct from other cohorts, most especially from the Tajima's D estimator, where we see that, well, the rest of the species, that is Anopheles arabiensis and Anopheles gambiae, they have a negative Tajima's D. We see that the Pwani molecular form has a positive Tajima's D, which is an evidence of a recent population bottleneck, whereas a negative Tajima's D is an evidence of population expansion. And so we see that this this unknown species, that is the Pwani molecular form, has a very different demographic history from the present species, that is Anopheles arabiensis and Anopheles gambiae within the country. So this also solidifies our evidence that there is a presence of a new cryptic taxon, which is a taxon, which is the first to be recently identified in East Africa, given that the three most recent cryptic taxa were identified in West Africa. So we went on to do also the insecticide resistance analysis, and here particularly we looked at the known genes that are implicated for insecticide resistance. And we went on to also look at genome-wide selection scans to look for any look for any mutations or areas that might be implicated in insecticide resistance, but were previously unknown. And we also went on to look at how far the spread of resistance within and across species, as well as across geographies. So the first analysis we looked at the single nucleotide polymorphisms within markers within the target site gene, and we see we particularly looked at at three main genes, that is the VGSC, the RDL, as well as the ACE1. Mutations within the VGSC have been mutations within the VGSC, such as L995S, have been previously known to be associated with resistance to DDT as well as pyrethroids. And we can see that in the VGSC, we have mutations on the L995S mutation in the Anopheles gambiae populations, both from Muheza and Muleba, despite them being of different frequencies. We see the Anopheles gambiae from Muleba has a very high frequency of L995S, whereas the Anopheles gambiae from Muheza has a moderate frequency. And this might be associated with the fact that the Muleba area is known to have been an area which has had high malaria prevalence. And so many interventions were firstly deployed in that region. And so there has been like a use of insecticides for a very long time. And so many mosquito populations in that particular area might have, actually more exposed for longer periods of time to insecticides. And so this may likely be the source of like the resistance mutations that we see in this particular population. The next mutation that we see is the I693V, which is only in the Anopheles gambiae from Muheza, but this particular mutation has not previously been known to be associated with resistance in any of the populations in the country. And we also see there is some mutations in the RDL, that is the V327I and A296S, in the Anopheles arabiensis populations from Moshi. And these mutations have actually been associated with resistance to dieldrin, which some research from the same area has identified that this particular mutation may be associated with the usage of agricultural pesticides in these areas, given that there is only the only insecticide intervention for malaria control that is used in this area is the pyrethroid impregnated long-lasting insecticide-treated nets. And so the likelihood that this particular mutations is coming from the malaria control interventions is very low, and therefore this may be due to the previous use of, the use of like agricultural pesticides, which might have which may likely be leading to these particular mutations. And in the ACE1, we see a number of mutations, but particularly in the Pwani molecular form from Moheza. But many of these mutations have actually not been previously known to be associated with insecticide resistance. And so further studies might be useful in terms of determining whether these mutations are associated to any insecticide resistance in these populations. We went on to look at also the copy number variants in the metabolic, that are associated with metabolic resistance in cytochrome P450 genes, as well as the carboextrase gene clusters. And we see that there is a very high frequency of CYP6AA1 and CYP6AA1 in the Anopheles gambiae from Moleba. And it's also coupled with the CYP6AA2 in the same population. And these have been actually previously associated with resistance in pyrethroids. And this actually coincides with what we see from the target site resistance analysis, the SNP analysis, where we see that there is also a very high frequency there is a very high frequency of L995S, which is associated with pyrethroid resistance. And so this shows that this population is likely to be, this population is very highly resistant to pyrethroid. And on top of that, we also see there is a CYP9K1 resistance at 81% frequency in the same population, which also just confirms the presence of high resistance in this particular population of pyrethroids. And on top of that, well, we don't see any other mutations in the cytochrome P450s within the Anopheles gambiae from, Anopheles arabiensis populations. We see a moderately high presence of frequencies in the carboextrase genes cluster within the Anopheles arabiensis from Moshi. This particular gene has been recently associated with the presence of resistance to primus methyl, but this study was done in the West Africa, and so the likelihood that we see this mutation here might be due to the same. And we also see, while we don't see any presence of copy number variants in the cytochrome P450 genes in Anopheles arabiensis, a recent study with more recent samples or collections from Moleba and Moshi have shown that there is like some high frequencies in the in the cytochrome P450 genes within these populations that were collected from the same location. And so we went on also to look at the genome-wide selection scans, and from this we see a peak at the CYP6AAP gene cluster, which is associated to to pyrethroids across all the population cohorts. We also see a peak at the VGSC gene in the Anopheles gambiae from Moleba, which also coincides with our results that we have seen from the SNP analysis earlier. And we see a a peak also near the carboextrase gene cluster, which we have also seen in the previous analysis from the copy number, from the copy number variant analysis within the population of Anopheles arabiensis. And this just coincides with the results that we have seen previously. And this shows that most of these populations are implicated with the presence of resistance in pyrethroids. And we went on to look at the haplotype networks, which actually show how much the resistance has spread within and across species as well as geographies. And the first panel here is an analysis of the same, but we were looking at the spread of resistance haplotypes within and across different species, that is Anopheles arabiensis, gambiae, and the Pwani molecular form. But when we look at this particular network here, we see that it's only shared with within the Anopheles arabiensis, particularly the majority, whereas this particular cluster is shared between two species, that is Anopheles arabiensis and Anopheles gambiae. And we have another small cluster here, which is which includes haplotypes that are only shared within the Anopheles gambiae. But when you look at this shows us that given that there is a shared haplotypes of resistance between two different species, this may likely show that there is a presence of like a spread of resistance across species. And we went on to look at like the spread of resistance within species, but across geographies. So this first one is actually for Anopheles gambiae, and within the Anopheles gambiae, the first one is for Anopheles arabiensis, and within the Anopheles arabiensis, we see there is a shared haplotypes of resistance amongst from different different locations. So this shows that there might be a spread of resistance within this species across geographies, which also coincides with our results that we have seen that Anopheles arabiensis actually cluster together. And so this shows us that there might be no restriction of gene flow, including resistance genes or resistance mutations. And for Anopheles gambiae here, we see there two separate clusters. The first one only includes only has Anopheles gambiae only shows the a shared haplotype, resistance haplotype within the gambiae from Moheza, whereas another one from Muleba, which also coincides with our results that we have seen that there is a geographical separation between populations of this particular species. And the last one is actually for the Anopheles, for the Pwani molecular form, which does not tell us much, but it looks like we don't see any, we see like insecticide resistance haplotypes, but they are not very major. So to conclude, our study has shown that there is a new cryptic species in the coast of Tanzania and Kenya, and this is we've named it the Pwani molecular form. And this particular species may contribute to residual malaria transmission, especially in the coastal areas, as it only exists in the coastal areas, and so the likelihood of it being a saltwater species is very high, and saltwater species like Anopheles maris have known to be contributing to residual malaria transmission during dry season. And then we also see a population structure between Anopheles gambiae from the east and the western side of the country, and this shows us that this, as we have said earlier, that the Anopheles gambiae might likely be due, the population structure within the Anopheles gambiae might likely be due to the physical adaptation, given that it prefers only humid areas. And so given that the middle part of our country is semi-arid, or it's characterized by semi-arid or arid climate, then the likelihood of Anopheles gambiae from the western side and the eastern side of the country to be separate is very high, whereas it's different for the Anopheles arabiensis populations, which are homogeneous, and this is also due to their adaptability to different climatic conditions. And we also see the presence of pyrethroid caridia resistance in Anopheles gambiae populations, but not in Anopheles arabiensis. And we also see carboextrase CNV-mediated resistance to pyrimidine methyl in Anopheles arabiensis from Moshi. And lastly, we also see resistance haplotypes are possibly spreading not only across geographies, but also across species. Lastly, I would like to thank Dr. Alistair Miles, Dr. Kelly Bennett, and Professor Pedro Okumu, who have been very supportive of this work and in terms of like my training. And I would also like to thank the malaria gen data contributors from different parts of the from different parts of the places that we have used the data from, like Kenya, Uganda, and Tanzania, as well as Dr. Sanjay Nagy, Dr. Dave Whitman, and Professor Martin Donnelly from Liverpool, Dr. Francesco Baldini, and Dr. Simon Babayan, who are my PhD supervisors, who have also been supportive of this work. The Bill and Melinda Gates Foundation, Ifakara Health Institute, and the Malaria Gen, as well as the PAMCA for training and financial support throughout my PhD. Thank you.
Okay. Thank you so much, Sophia. That was fantastic. Congratulations to you and your co-authors on this discovery. We've now reached time for Q&As. Kindly raise your hand or type your question in the Q&A box, and I'll read them for Sophia and she'll address them. I see there's a question already from while you were presenting. Professor Deasi Shongoma asked, what were the study sites in Kenya?
In Kenya, there was only one study site, that was Kilifi.
Thank you so much for that. We have time for Q&A, as I say, so please type in the chat box or raise your hand. Dr. Jolo Dero says, great work, Sophia. There are two questions. From your metadata, how were the Pwani molecular form mosquitoes collected? Were they in or outdoor collections, larvae or adults? So that's the first question. Do you have a DNA or PCR-based assay for field monitoring of the new molecular forms? So those are the two questions from Jolo Dero. Sophia?
Okay, so the data from, the Pwani molecular form samples were collected using, I said earlier here, using pyrethroid spray catchers as adults, and they're collected indoors. And we do not have DNA-based assays for field monitoring of the new Pwani molecular form yet, I think. But I think Alistair or Kelly, if they're in the room, they probably would be able to know better about the DNA-based assays, if we have them or not. So, yeah.
Thanks, Sophia. And just to say, Kelly is in the room. So Kelly, if you do want to say something, kindly allow us to unmute you and you can say something. But there's a question now from Dr. Richard Pearson, who says, really interesting talk. Thanks, Sophia. It looked like the FST between Pwani from the two locations was higher than between Pwani and Anopheles gambiae. Isn't this surprising given their relative positions on the NJT? That's from Richard Pearson.
Between Pwani from the two locations was higher. The likelihood that it was higher between the two population, I mean, between the same population, I mean, the two unknown species is likely maybe also contributed to the geographical distance. Yeah, I think so. I'm not too sure, but yeah, I think that might contribute to a slightly higher FSD value.
Thanks, Sophia. The questions are coming in and comments as well, so I'll read a few. We still have about 15 minutes to go, so please keep the questions coming in. There's a comment from Dr. Kelly Bennett, who says, hi, just to add that we do not have plans for an experimental molecular-based assay, but by comparing sequence data to samples from East Africa, identification of molecular form is possible. So that's responding to the question by Dr. Odero earlier and adding to your answer, Sophia, from Dr. Kelly. We have a comment and a question. There's a comment from Salem Gudo, who says, hello, Sophia and team, this is great work, well done. I'm Salem Gudo from Kenya. I would like to let you know that I did my MSc project on malaria elimination using botanical insecticides from mangrove plants as synthetic insecticides are facing resistance. If given the opportunity to work with your team for a PhD level, I would really appreciate, if any. That's from Salem Gudo. I don't know if you have any comments to that, Sophia.
No, but I think I'll reach out.
Thank you, Sophia. And there's a comment from a question and comment from Professor Fredos Ukomu, who says, nice presentation, Sophia. To ensure a comprehensive understanding of the newly identified cryptic mosquito species, would it be possible to expand your sample collection to include additional regions, such as the southern, western, as well as central parts of the country? This broader geographical coverage could help determine whether the species is present in areas where samples have not yet been collected, providing a more complete picture of its distribution. That's from Professor Okomu. Sophia?
Yeah, I think, yes, that would be a very good study to give more understanding in terms of like the distribution of this Pwani molecular form, given that our study was only restricted to 4 locations, which does not really capture the entire picture. So like, of course, a further study that would include all these locations that have been suggested by Fredros would actually really be useful to the entire study.
Okay, thanks, Sophia. There's another question from Professor Ishengoma. Congrats, Sophia, great work. Can the population structure in Anopheles gambiae from Muleba versus Muheza be due to a recent bottleneck as we are seeing a decline in the populations in Tanzania? Our recent data shows that Anopheles gambiae is less than 2% of the vectors we collected in 2023. That's from Professor Ishengoma. Sophia?
I'm not too sure, but maybe it might be likely, but I'm not too sure about that. I can't really say the recent population. I mean, the recent bottleneck or population bottleneck might be contributing to the population structure we see, but I personally think what really causes the population structure in Anopheles gambiae is actually the climatic condition and the presence of the Rift Valley, given that we know the characteristic of Anopheles gambiae as a species, as the species themselves, they're like they like humid areas, and in the middle we see that there is a semi-arid or arid area. So maybe the population bottleneck might be an additional factor, but yeah, I can't say it with certainty.
Thanks, Sophia. I see Professor Ishengoma is in the room. Prof. Des, do you want to say something?
Yeah, I just wanted to say we are seeing similar patterns in drug resistance between Muheza and Moshi, where we have high rates of molecular markers of SP resistance. So I was expecting, actually, if there are gambiae, which I know in Moshi, not common to see gambiae, they shouldn't be separated. They should be homogeneous. But again, if Arabienses are homogeneous throughout the country, there should be something else which is happening within the gambiae population. So maybe some more work. So great job. And we do have more samples from Muheza 2023, and we are collecting more this year. So we should collaborate and do some more analysis to see what we are able to get. And we can arrange some more collections down in Ilindi and in Mtwara to see whether it's along the coast all the way down towards Mozambique. But a great work and great presentation. So congratulations and best wishes in your PhD.
Thank you.
Okay, thanks, Professor Shangoma. There's a question from Dr. Polo, Brian Polo, saying, great work, Sophia. The question is, why do you think that the Pwani molecular form does not have any resistance markers? Sophia?
Okay, so I personally think it's because also our sample size was a little bit smaller to identify whether there was any resistance markers. So like a bigger sample size would have been more useful. Yeah, I think that's the main reason, major reason why we couldn't really identify whether there are any resistance. Like we saw some resistance markers, but we couldn't really tell whether they were, what were they associated to, like when we looked at the copy number variants in the carboextrase, I think that was carboextrase gene. Sorry, just go back to that. No, yeah, it was the SNPs in the ACE1 gene. We see some frequencies, but we can't really tell like what these mutations were associated to. Yeah, but it's also because we really have a very sample size in of this particular unknown species in Tanzania.
Thanks, Sophia. I think it links nicely to the question by Professor Okomu as well about perhaps expanding the region of the study as well. We still have about 10 minutes to go, so please do raise your hand or type in the chat box. Another question from Dr. Richard Pearson. The H12 selection scan showed a peak at CYP6AA for Anopheles arabiensis from Muleba, but I think there were no high levels of resistant markers in this population in previous slides. Is that right? Does this mean that it is unclear exactly which mutations are driving this peak? That's from Dr. Pearson. Sophia?
Sorry, can you say that again?
Okay, I can read it aloud. The H12 selection scan showed a peak at CYP6AA for Anopheles arabiensis from Muleba. But I think there were no high levels of resistant markers in this population in previous slides. Is that right?
Yes.
Does this mean that it is unclear exactly which mutations are driving this peak?
Yeah, correct. From the study, it was unclear, given that we see a peak, but we don't see any mutations in this particular area when we do the SNP analysis. So yeah, it could be that they were driven by something else, but then a recent study by Eric Lucas identified that there was copy number variants in that particular region. So.
Okay. Thanks, Sophia. I'm gonna give you a few more minutes. We have about 8 minutes to go. Please type your questions in the chat box or raise your hand, and Sophia will gladly engage with you. Sophia, great presentation, as everybody has been saying. I'm just wondering, what do you think are the implications for malaria elimination in Tanzania and Kenya with the sort of discovery of Pwani molecular form? Do you think it's gonna regress the progress or make you find a solution faster or easier? What do you think is the implication of this discovery?
I think it's both. It could help in finding the solution faster, given that we already know that there is a presence of something that was previously unknown. And I can't really tell, because when we look at our data, we haven't really looked at the vectorial capacity of this particular unknown species. So we can't really tell whether it does spread malaria or not. And so further studies might be more helpful and useful in terms of determining, in terms of determining whether this unknown species contributes to malaria transmission or any other, infectious disease.
Yeah. Thanks, Sophia. Okay. I'm looking at the Q&A box and looking at the chat box as well. I'm giving you one more go for questions or comments. Going once. Going twice. Okay. Any last comments from yourself, Sophia?
No, not any from me.
Okay.
Thank you, everyone.
Well, thank you so much. This was fantastic. Congratulations, indeed. And I think I've been inspired by your work over the past few years, and collaborating with you has been amazing. So congratulations. And yeah, hope you and your team can discover more species across Africa and help us eliminate malaria in our lifetime. To those of you that attended today, thank you so much for joining us. This General Club was recorded and will be available later today on Malaria Gen YouTube channel. Kindly share with your networks and colleagues and those of your friends that couldn't attend today. We are going to have another General Club sometime in March. Be on the lookout on the Malaria Gen GSU LinkedIn as well as X pages, as well as Blue Sky as well. And you can join the newsletter to receive announcements about our future events as well. Thank you so much for joining, everybody, and see you next month for a different General Club. Thank you so much.
Yeah.